What I did tell you last time is the general apparatus that is used by the plant, all right? And I showed you also a structure of where exactly the light harvesting, etc., takes place, all right? And what I did mention briefly last time is that that there are two sides, in the plant, in the leaves, where actually light is harnessed. Okay, one is at a slightly different, longer wavelength than the other. So this actually absorbs around 700 nanometers, whereas this absorbs around 680 nanometers. Okay, so this is the light that is falling, okay, on the chloroplast in both these cases. Okay, but they basically perform somewhat different functions. Remember that the longer is the wavelength, it means the lower is the energy. Okay, so this is absorbing light of a somewhat higher energy than in this particular case. Okay, and what these do is that they produce some of the primary chemicals, which are high energy products, which are actually used subsequently in carrying out some of the reactions, because many of these reactions actually require energy. And so you don't have a Bunsen burner which you heat with or apply electricity. Okay, so plant does it in a different way. It stores the energy as high energy products. Those high energy products release energy and get converted into low energy products. Those low energy products again come to the site of photosynthesis and again become high energy products. Okay, so that's really what the light harvesting portion does. So let me try and explain to you what first. Let me first come to photosystem 2. Now, photosystem 2, as you can see, it's basically absorbing the light, and whenever you absorb light, when a compound absorbs light, basically the electrons move into the excited state. Okay, so they are at a higher energy. So they move from a certain orbital which is at a lower energy to another orbital which is at a higher energy. Okay. Now, what happens is that, you see, if, for example, if you just don't do anything to it, like you can take, say, chlorophyll or something and just make an aqueous solution, and pump it with light, nothing will happen really. Okay, or there are so many molecules that absorb light, but all that will happen is that the electron will climb to the higher energy state, and then after that it will just relax back to the lower energy state. Okay, I mean, it may release the heat, or it may fluoresce or luminance, and may release it in the form of light, but the two will tend to recombine. All right, the electron and the positive charge that you created. Okay, but in the plants have created a wonderful mechanism by which what it does is that the moment that the electron is formed in this higher energy state, it gets swept away, right? And because it gets swept away, right, that electron cannot now combine with the holes that it left behind, right? Now, and so there is a positive charge that gets created here, and there is a negative charge, and they have now got spatially separated, and they really cannot again combine. All right, that is what nature has evolved over millions of years, perhaps, and so many of the people would like to try and replicate something like this in the laboratory, but it's extremely hard, to copy nature. Okay, nature system is way too complex. Okay, I mean, people have done things, but nothing, of the kind that nature is able to do. Okay, so now what happens is I've got holes here, right? And hole means that it is wanting an electron. That means it can be looked at as an oxidizing agent. It has got the ability to oxidize something. And so what happens is that these holes are basically seen by an enzyme which contains manganese. It's a manganese containing enzyme, all right? And that enzyme has the ability to break up water, okay, and release oxygen. So hydroxide ion from water would get oxidized, and oxygen would be produced. That's the oxygen, half O2. So oxygen is coming out, right? So as you know, in photosynthesis releases oxygen. Okay, so that is taken care of by formation of oxygen. Now the electron, as I told you, it basically starts doing like a relay race. Okay, and there are many of these compounds, which can effectively take electron, get reduced, and again get oxidized, get reduced, and the electron is sliding down to a lower energy state, as it is moving there. Now, over here, something very interesting happens, that the electron basically obviously has an energy, okay? So it can give off energy. Now that electron, it gives up its energy to ADP, okay, which is adenosine diphosphate, okay, and ADP in the presence of H plus, which is present here, and in the presence of phosphates, which are also present, it gets converted into ATP, which is adenosine triphosphate. Okay, and ATP has a higher energy than ADP. So you took a lower energy compound and effectively converted it into a higher energy compound, and that energy we will subsequently require, we will use, and show you where we are using it. Okay, and what happens here in the photosystem 1? Same thing as here. It absorbs light, the electrons go into the excited state. Now in this particular case, what happens is that the electron which migrated from here, okay, which only gave off its energy, I did not give off the electron as such to ADP, I only transferred energy. So the electron basically now goes and combines with the hole which is formed here, and basically takes care of that, right? Okay, and the electron which was generated over here in the excited state, that electron, exactly like here, it moves again, it does a relay race, and it goes through a number of different kinds of enzymes and proteins. Okay, things like iron sulfur proteins, ferredoxin, etc. And again, it is getting spatially separated. Okay, and the electron is climbing down. Now, over here, you have another compound, and these are the two fundamental things which are produced in photosynthesis besides oxygen. This is NADP, okay, and I'll show you what the structure of NADP later on. NADP is in an oxidized state when it comes here, and in the presence of protons, okay, and in the presence of the electrons, it gets reduced to NADPH. Okay, that is NADPH. Okay, so NADPH is a reducing agent because it got reduced, and now it can go and reduce something else, and in turn can get oxidized back into NADP plus. Okay, and again it will find its way into the photosynthetic side, and again get converted into NADPH. So that's effectively what's happening. So you ended up with two high energy molecules, ATP. ATP's sole responsibility is really to make energy available, okay, for a process. And NADPH is in a sense also supplying energy because it's at a higher energy level, plus it acts as a reducing agent. So NADPH serves two purposes. It's a reducing agent, plus it also supplies part of the energy. And what else did we form? We formed oxygen. Okay, and that is the primary photosynthetic process. Has everybody understood photosystem 1 and 2?
Yes, sir.
Okay. Okay, now let us quickly go through the structures of ATP and ADP. Okay, now if you look at, this is the adenosine, compound. Okay, and that compound is the same here. There's no difference. Okay, identical. Okay? What happens is that in ADP there are two phosphate linkages, so it's a diphosphate, okay? But in ATP, one more phosphate joins, okay? And it becomes a triphosphate, okay? And that phosphate comes from this HPO4 2 minus, and in presence of H plus, it basically forms the triphosphate. Now, the third phosphate, when we put in, it pushes the molecule into a higher energy level. So ATP has got higher energy than ADP. Okay. Now, how much higher? It is when ATP gets converted to ADP, it releases 7.3 kilocalorie per mole. Okay, not a very high energy release, but it's still substantial. And nature has got a beautiful trick where it combines the energies of several of ATPs, and gets quite a bit of energy for its processes. Okay, it's not just one ATP. You may require 6 7, okay, ATPs, or 9 ATPs can get generated in a process. It's very interesting. Okay, so that's really the chemistry of ATP, ADP going to ATP. That's all that is happening, in this over here. Okay, in this part. Okay, that's all that is happening. ADP came in and ATP went out. Okay. Now, similarly, NADPH, okay, and NADP plus, right, these are very interesting. This top one is NADP plus, okay, this one. Now you just concentrate on this part of the molecule, okay, you will see that it's an aromatic system, right? With three, it's like a pyridine derivative, okay, and it's got aromaticity here, okay. Now when that H plus and the electron is going and reacting with the NADP plus, it breaks the aromaticity, okay, and it forms this. This is known as NADPH. This part of the molecule is otherwise the same, okay. There's no difference. Like all of this is more or less the same, okay. What is different is only this part. Okay? This became this, okay. Nothing else is different. But this is a good reducing agent, so it can transfer hydrogens, right, to other molecules, and again get converted back into NADP plus. Okay. Now let us look at the energetics of this process. NADP plus, which is this, requires 2 electrons plus a proton, right, to get converted into this. Okay, so this is a 2 electron process. Okay, going from here to here. Now the redox potential for this is equal to, for this reaction, is equal to minus 315 millivolt. What does that mean? Is it a spontaneous reaction or is it a non-spontaneous reaction? Is it a thermodynamically favored reaction or is it a thermodynamically unfavored reaction? This reaction. are you aware of the uh, um, um, relationship delta G is equal to minus NFE?
Yes, sir. Gives free energy.
Right. Okay. So if my E is negative and I've got minus NFE, so my delta G will become positive.
Yes, sir.
Right or wrong?
Yes, sir, it will become positive.
Right. So if it becomes positive, what does it mean? When delta G is positive,
It is not
Not
in,
favorable.
not important.
so it's a non-spontaneous reaction. Okay. So I mean, you just have to go to the basic equations like delta G is equal to minus RTLN delta G is equal to minus NFE, all right? And you'll come to know, right, what it is. Okay. Now, so that's what this is. Now, how is it then happening? It's happening because the electrons are coming here at a high energy level. It's in an excited state. Okay. So the extra energy that it required to actually carry out the process, it came really from the electrons which were moved to the excited state from in the photosystem 1. All right. So that electron in a higher energy state now has the required energy to overcome the non-spontaneous nature of the process. Now, delta G is equal to minus RT ln K, that's also a relationship, or I can relate E with K because delta G is equal to minus NFE and delta G is equal to minus RT ln K. So I can write minus NFE is equal to minus RT ln K. So I have a relationship between E and the equilibrium constant K, right? So obviously, depending on these concentrations, I can actually work out what will be really the actual potential for actual conditions of use where you can define how much of NADPH is there, how much of NADP plus is there. Okay, so effectively that's how you relate these things. Okay, and but the most important thing for you to remember is that both of these processes of ADP going to ATP and NADP plus going to NADPH are thermodynamically non-spontaneous, and they are actually taking place by utilizing the energy from the sun, okay, which pushed electrons into a higher energy state, and that is what is supplying the energy for these processes. Okay, now let us. So what happened? I created ATP and ADP, right? And once I've created ATP and ADP, its role is pretty much over. Okay, and the oxygen gets released by the plant, and it builds up in the atmosphere. Okay, so then what happens is the plant now says, oh great, I have got a source of energy. I've got ATP and I've got NADPH. Okay, and there is a dark cycle which does not require light, and that's where some of the molecules are getting synthesized. Okay, so let me show you. And getting synthesized from what? Because plants generate carbon containing molecules, so those molecules are being synthesized from carbon dioxide. That's why we know that plants take in carbon dioxide and release oxygen. So now let us try and understand what's going on. Now, one of the things for you to remember, which is the big difference between like the way we do reactions in the laboratory, and what is done by a plant or an animal, okay, is that plants and animals have enzymes, which are like the catalysts which promote the process. Okay, and so that's what enzymes are doing. Enzymes are mediating these reactions. Like for example, in the laboratory, if you had to just take carbon dioxide and convert it into sugar, it would be next to impossible to do. We do not know how to do it. Okay? Nobody has figured it out. But plants do it effortlessly. Okay? So let's try and explain, all right, some of the basic chemicals, okay, and what's going on. Now, what is going on is that, as I told you, ATP and NADPH moved into the space where the dark reaction is taking place, and they are saying, we will supply you the energy, and the reducing agents, don't worry. Okay, so we start with this particular enzyme, which is known as ribulose bisphosphate. Okay, it is a 5-carbon molecule. So you can see 1 carbon, 2, 3, 4, 5. It's got 5 carbons and it has got 2 phosphates. That's why it's called bisphosphate. Okay, so this stuff, 3 of these plus 3 carbon dioxides, they come together. Okay, and this ribulose does something remarkable which otherwise we struggle to do with organic molecules, that the carbon dioxide comes and binds quite strongly. Of course, carbon dioxide will also bind strongly to sodium hydroxide. I can react sodium hydroxide with CO2 and it will form sodium carbonate and sodium bicarbonate, but it's not going to be so easy to release it or to convert it into something else. Okay, but nature does that. Nature maintains a very subtle balance between these molecules and is able to delicately convert one molecule into another. Okay, so now I took 3 of these and I took 3 of these. So the total number of carbons is 3 times 5, 15, and 3 CO2s. So 15 plus 3, 18. Okay, so I've got 18 carbons which are involved in the primary process of fixation of carbon dioxide. Okay, so the CO2 comes and binds here. Now, when the CO2 comes and binds, this molecule, falls apart. Okay, and in turn, it generates 6 molecules of a 3-carbon entity. So from a 5-carbon entity plus 3 CO2, that is 18 carbons in all, I have generated a 3-carbon entity. 1, 2, 3, and 6 of those. So there is mass balance, okay, of carbon. All right, 16, 18, and 18. Okay, what is this molecule? This molecule is 3-phosphoglycerate. Okay, now I have shown you the structure for 3-phosphoglyceric acid. So all you have to remember is that it's an ester. Okay, instead of the acid, you form a ester. Otherwise it's identical. 3-phosphoglycerate. So there will be a ester formation. So it will be OR. That's all. Okay. Now, this over here did not require any energy from outside. This molecule is at a high energy level. So it has sufficient energy to actually carry out this conversion on its own. It doesn't need help from anybody. Okay, it doesn't require any energy source from elsewhere. Okay, but when this is formed, right, this reacts with NADPH and the acetate, the ester group, this over here gets reduced to an aldehyde. This is an aldehyde, right? CHO. The rest of the molecule is the same. You can see phosphate with the hydroxy group over here also there. Only thing is that the carboxylic acid group got converted into an aldehyde. So it needed a reducing agent. Okay, that reducing agent is basically coming from NADPH. Is it all that is required? No. It requires quite a bit of energy. It's an uphill conversion. Okay. So in the process, 6 ATP molecules now get converted into ADP and release its energy. Okay, that's 7.3 kilocalorie per mole. So you can multiply 7.3 times 6, that many kilocalorie per mole to convert 6 of these phosphoglycerates into phosphoglyceraldehyde, this molecule. Okay, that's all that happens. So the step 2 is reduction of 3-phosphoglycerate to G3P, which is glycerol 3-phosphoglyceraldehyde. Okay, that's effectively this one. All right. Now, what is also very interesting is that the moment, one more thing. Now, this thing you can see, Calvin was awarded the Nobel Prize for the C3 cycle. Okay, and we will encounter another cycle later on, which is known as the C4 cycle. So let me tell you what is C3, where the name comes from. The first stable molecule that you produce, in the course of photosynthesis, right, is the number of carbons in that is what is meant by CX, C1, C2, C3, C4, whatever you want to call it. Now, the first stable molecule that is being formed here is really 3-phosphoglycerate. It's a fairly stable molecule. Okay, it is just that in presence of ATP and NADPH is getting further reduced, but that doesn't make the molecule unstable. The molecule is quite stable. Okay, so the first stable molecule that you produce from photosynthesis and the number of carbons that it has determines what you call it in terms of the cycle number. And in this particular case, it is called a C3 cycle because this molecule bears 3 carbons. Okay, so it's a C3 cycle. So now we have gone through the second step, which is the reduction of the 3-phosphoglycerate to the aldehyde over here. Now nature plays another very brilliant trick. What it does is that it keeps aside, one of these molecules out of the 6, one it keeps aside. Okay, and this is later on converted into glucose, fructose, etc. Okay, so it's kept aside one, and the remaining 5, what happens is that this stuff, okay, combines together. Now you've got how many carbons? 5, and each one has got 3 carbons. Okay, 1, 2, 3. So 3 carbons multiplied by 5 molecules means 15 carbons. Okay, so those 5 go and again reform ribulose bisphosphate. Okay, this, and it will form from 5 G3P, it will form 3 RUBP, so that you conserve the carbons. 5 times 3 is 15, 3 times 5 is 15. Okay, this again requires energy. Just like this required energy, this also requires energy. How much energy? 3 ATP is getting converted into 3 ADP. Okay, so in all, I have consumed 9 ATP molecules and 6 NADPH molecules to form one glycerol phosphoglyceraldehyde, which will basically be equivalent to half a glucose molecule, because glucose is a C6 carbon. Okay, and this is C3. So it's like saying that I need 2 of these, to form glucose and fructose and things like that. Okay, so in that case, obviously I will require 2. For 2 of these, I will require a total of 18 ATP and 12 NADPH. Okay, to make 1 molecule of glucose from 6 molecules of carbon dioxide. okay, because I need twice that amount to form glucose. Okay, so fixation then is 3 RuBP plus 3 CO2 going to 6 of the 3 phosphoglycerate, and that gives it the name C3 cycle. 3 phosphoglycerate, 6 of those, in presence of 6 molecules of ATP and 6 molecules of NADPH, get converted into 6 molecules of this one 3-phosphoglyceraldehyde, of which 1 is utilized to produce glucose and fructose, and 5 gets recycled back in the process into this. Okay, and that is really the dark cycle of photosynthesis. Okay, and all these, effectively take place in the stroma of the chloroplast. So it's here. This is how it looks. Right? Okay. So you can see that in the dark reaction, if I had to convert, let's say if I want to make a fructose, okay, so fructose 6-phosphate, not a pure fructose, but with a phosphate attached to it, okay, I would require 6 carbon dioxides plus 12 NADPH2 plus 18 ATP goes to this, and again forms the low energy versions of these compounds, and these will again find their way into the light harvesting region and get regenerated back into NADPH and ATP, and the entire process continues. Okay, and this is how conversion of 200 billion tons of CO2 to biomass. That's what's happening. This is the main reaction that takes place. Okay, so I hope you understand where your biomass is coming from, the fundamental processes involved. Now, let us try and go a little more into a few more details. Okay, the gist of it I've already explained, but let me still tell you that there are subtleties, okay, which you must understand. Now, firstly, the affinity of Rubisco for CO2 is high, and it's a spontaneous process. It did not require any other energy source, so it must have been a thermodynamically favored process. Okay, and it undergoes a rapid carboxylation, okay, and it then gets converted into the 3-phosphoglycerate. Okay, breaks apart. First takes in the CO2, okay, and binds strongly, and then falls apart, and forms 5 of those, 6 of those molecules. Okay, now the negative change in free energy associated with carboxylation of RuBP is large, so the forward reaction is favored. It's saying the same thing. Okay, so in a way, the kinetics and the thermodynamics are both pretty good, because the actual concentration of CO2 is quite low. You know that the concentration of CO2 in the atmosphere is very low. We are talking about 300, 400 ppm. Okay, so under those conditions, for the molecule to be able to bind the CO2 and then react, obviously shows what an outstanding affinity it has for the CO2. So the moment the CO2 is swept away, the concentration gradient will continuously allow more and more CO2 to come into the site of the dark cycle. Okay, but remember also that Rubisco, all right, is not just having an affinity for CO2. Rubisco can also react and interact with oxygen. So you need to be very careful, okay, in being able to keep the oxygen levels quite low. So obviously the place, in the stroma where these things are going on, there must be a very good mechanism by which the oxygen is more or less kept excluded. But all of the oxygen is not shut off, okay? And so the oxygen also reacts with Rubisco, and that reaction is known as photorespiration. Okay, so this is what is allowing photosynthesis, and this is what will allow photorespiration. Okay, and the process of photorespiration will generate carbon dioxide. Okay, any questions? Is everybody understanding?
Yes, sir.
Yes, sir.
All right. Now let us look at another thing which is very interesting, and I'm showing this only because we have discussed a little bit around the isotopes. When we were talking about atmospheric carbon dioxide, and we were talking about signatures, right? Like we said that fossil fuels do not have any carbon 14, so that when they're burning, there's no signature of carbon 14, whereas a live plant, will continuously also take in some amount of carbon 14 because it's getting generated. Okay, and we saw the pathway by which carbon 14 is formed. Okay, now so in the atmosphere, what you have is, of course, I have tried to show you only the carbon 12 and the carbon 13, but there is also some amount of carbon 14 which is present in CO2. All right. So all of those are pretty much there. But over here, I've just tried to concentrate on the carbon 12 and the carbon 13 because the carbon 13 is not so low. It's 1%. It's 1% by weight, okay, in the atmosphere. Now, what is very interesting is that, between carbon 12 and carbon 13, which is heavier?
13.
13, sir.
Okay. Now, usually we know that the heavier the molecule, the more sluggish its motion becomes. Okay. So what happens is that when the carbon 12 and the carbon 13, after all, how does it get into the plant? It gets in, through the leaves, and it has to diffuse into the leaves, right? So what is found is that carbon 13 tends to diffuse slightly slower than carbon 12. So naturally, inside the plant, you'll have relatively more of carbon 12 than carbon 13 as compared to what is there in the atmosphere. Okay? Second, what's been found is that carbon 12 tends to react with Rubisco a bit more efficiently than carbon 13. Okay, so effectively what's happening is, and those are the carbon dioxide which is ultimately becoming your sugars and stuff like that. Okay, so what you'll find is that the sugars etc will have a little more of carbon 12 and a little less of carbon 13 than what you will have in the atmosphere. So that becomes a signature for a plant. And so when, for example, let's say you are burning wood or something like that, and again releasing CO2, that CO2 which will come out, we will have a different signature from the atmospheric CO2 or from the fossil fuels signature. Fossil fuel, well, in a way, yeah, this one will have also the carbon 14, which I didn't talk about, which would have disappeared. The carbon 12 and carbon 13 may not be so different in the fossil fuel because, after all, what is fossil fuel? It is the same carbon which was buried, and it's not radioactive. So it was buried and gradually got converted into carbon, and fuels. Okay. So the carbon 12, carbon 13 ratio may not be so different from in a live plant, but the fundamental difference would be that in a live plant, in any sugar, etc., there would be also some carbon 14, which you will not have in the fossil fuel. Okay. So bear these in mind. And secondly, the atmospheric C12, C13 ratio is different, and so you may be able to, from the kind of changes that are taking place in these isotope ratios, you might be able to say, what is the macro situation in terms of photosynthesis, biomass getting converted back into carbon dioxide, etc. You may be able to get a handle of that. Okay, now let us look at a few more fundamentals, because we have seen how oxygen is coming out, we have seen how carbon dioxide is coming in, okay, and we know that you need light. And last time, we looked at how exactly the wavelength of the electromagnetic spectrum and the visible light spectrum is being generated. I showed you the equations, okay, and we also said that 680 nanometer and 700 nanometer are the places which where this will basically absorb. But what we have not said is anything yet about what happens if we increase the level of illumination. Like for example, I can have something in a very very sunny place, near the equator, or I can have it in a place which is more in the shadows, where you don't see much of light. So obviously we can understand that the level of photosynthesis will vary. I mean, qualitatively we know that, but let me tell you what is the quantitative behavior. Okay. Now, firstly, there is a point, okay, up to which, even if you have very little light, I mean, if there is some light, okay, but no photosynthesis will take place in this region, okay, and this is known as the compensation point. This is the point, at which you will get a positive photosynthesis. Now, the compensation point, why is it called compensation point? Can anyone tell me what is compensating what? Well, a plant is always also respiring, and the respiration process is producing CO2, So on the one hand, there is some CO2 being formed from respiration, and on the other hand, CO2 is being taken up for photosynthesis. When both of those are matched, I will have no net photosynthesis, no CO2 uptake, and so that will be my compensation point. Okay. After that, as I increase, the amount of light which is falling, okay, the irradiance level, okay, as it is increasing, this is my irradiance increasing, okay, what will happen is that my rate of photosynthesis will continue to go up, okay, and goes up. More light I put in, the faster and the photosynthesis will take place, okay, but it doesn't go on forever. Okay? It starts saturating, okay, so beyond a point it starts plateauing out, okay, and this is what is known as the photosynthetic capacity. After that, doesn't matter, even if you push more light in, the plant is going to say, I'm sorry. It's like, if I'm absolutely full and someone stuffs more food down my throat, I can't eat. Okay. So, now can anyone tell me what might determine the saturation?
Rubisco maybe.
Very good. In the sense that the dark reaction. Okay. Because you see, I mean, that's the beauty of the balance. ATP, NADPH, etc. is getting generated, going to the dark circles, getting consumed, coming back, and there is a lot of activity taking place. And because things are getting regenerated. But if I'm producing ATP, I'm producing NADPH, and if the Rubisco cannot take any more CO2 and convert it into phosphoglycerate, what's the point? I mean, it's going to slow it down, because what will the photosynthesis be there for? And already my NADPH and ATP are full. What will I do then? Okay, so I'll reach this kind of a saturation point. Okay, over here. Right? But you can imagine also that if my carbon dioxide in the atmosphere increases, then there's a possibility that my dark reaction efficiency will also go up. And not efficiency, but the actual quantum of dark reaction will go up. And so maybe if there is a little more of CO2, the saturation might happen at slightly higher levels. In other words, CO2 in the atmosphere could also be a good thing for photosynthesis, provided you have got enough of plants. But if you are cutting down forests and deforesting and causing desertification, doesn't matter if there is more CO2, but the plants are not there to take it up. And that's exactly what has happened now. I'm in an ideal situation to mop up more CO2 and cause more biomass generation, but I don't have the plants because I've cut down the forests. Okay, so that's the tragedy. We could have made better use of the CO2 if we had more plants. Okay, so this region over here where the photosynthetic uptake activity is increasing would be the light reaction limiting. All right, that means more light I put in, more photosynthesis will take place. It is this which is the limiting part. When I come here, it is being limited by my dark reactions, and so it is dark reaction limiting. Has everybody understood this graph? Has everybody understood
Yes, sir.
compensation point,
Yes, sir.
compensation level,
Yes, sir.
everything all clear? Okay, fantastic. Remember another thing that all plants do not behave in exactly the same way, right? So depending on species, there will be differences in this curve that I showed you, right? So for example, these are three different species of plants, okay? And what you can see is that they behave very differently. Like, for example, there are some, which will increase the photosynthesis rapidly with irradiance, but at a low irradiance level, and then after that it will rapidly plateau. There are others which will, maybe the slope is less, okay, and the photosynthetic activity goes on increasing with irradiance, and the saturation takes place at a higher level. So this is something like slow and steady wins the race, okay. I mean, that's what is happening with this, because you actually end up getting the highest level of photosynthesis. And then there are some which are poor in all counts, the irradiance also, even with irradiance, the photosynthetic activity is not high at this level, nor is it high in the dark level. And so, you get a pretty sluggish behavior, of this plant, all right? So the ideal in many ways would be really this red line. We should look for plants, which behave this way. Okay? That would be the best. But on the other hand, if you have got very low irradiance level, there are places like that where the light levels are very low. Okay, there the green might not be a bad idea. Okay. All right. Now I told you that Rubisco is also not just binding to carbon dioxide, but it is also binding to oxygen. Okay. So effectively, when that happens, it ends up forming carbon dioxide, and that is what is known as photorespiration. What happens? Okay, it breaks down, into 3-phosphoglycerate as in the previous case, but not with and to a 2-phosphoglycerate. Okay, and this stuff effectively releases carbon dioxide, this one, and so that leads to a destruction of the ribulose, because you have lost, some of the phosphoglycerate, okay, in the process. And this is what could happen if, for example, in the photosystem too, if a lot of oxygen is being generated, okay, and if the barrier between that oxygen and the stroma where the dark reaction is taking place is not good enough, then quite a bit of oxygen can get into the dark reaction side, and then these kinds of complications will occur, and it will be detrimental to the photosynthesis. Okay, the photorespiration might become a very dominant player. Okay, as compared to photosynthesis. Now, we'll see over here what kind of an effect it can have. All right, and this effect was actually discovered in 1920 by this German physiologist, plant physiologist Warburg. And he's the one who discovered it, and therefore it's known as the Warburg effect. Right? And what it means is that you can see if a plant, especially the dark portion, where the dark reactions take place, if there is too much of oxygen, okay, what you can see is for a fixed amount of CO2, if I started with a fixed amount of CO2, I maintained it, 275 ppm CO2, the rate of relative rate of photosynthesis, would be much lower when the oxygen increases. Okay, and this is true in at different levels of CO2. This is something interesting. But what is also very interesting is that photosynthetic activity has decreased, over a period of time. When I say period of time, I'm talking about millions of years, because initially the atmosphere did not have much oxygen. Today you have large amount of oxygen compared to carbon dioxide, but there was a time when the oxygen levels were lower. So as the oxygen increased, what happened is that photorespiration became more and more pronounced, and therefore the overall relative photosynthesis activity, the rate of photosynthesis, actually came down with the increase in oxygen in the atmosphere. So it was not all a good thing, but photosynthesis, too much of oxygen in the atmosphere can actually be detrimental. Okay, so keep that in mind. But now what is happening is, as we are burning more and more fossil fuel, what did we find? That the oxygen level in the atmosphere is actually coming down. Not coming down very dramatically, okay, but it is coming down. And the CO2 is going up. So again, what you will find is that the level of photosynthetic activity will start rising. Okay, where under these conditions, which is a good thing, provided you have enough of plants, to make good use of this. Okay? That's very important to mention. Okay, now plants have realized, that this is a problem. Okay, that the concentration of carbon dioxide going into the dark system is so low, because you are just taking it from the atmosphere, and so plants did something extremely clever. Okay, and the plants which are most important from the biomass perspective are those kinds of plants. Like, for example, sugarcane. Maharashtra has so much of sugarcane crop. It's Maharashtra and Uttar Pradesh are the two largest, in terms of sugar production. Okay, and sugarcane. Then, for example, maize, corn, okay, which is another very important crop, as you know, corn. Uh, Grasses. There are so many kinds of grasses that grow. Okay, sorghum. Okay, all of these follow a slightly different mechanism. Okay, and that mechanism was unraveled. Not, you can see 1966. We are talking about, not even just about 55 years. Okay, since these mechanisms were worked out. Okay, so these have not existed since, plants have existed since eternity, but the understanding is quite recent. Okay, it's just 50 to 70 years. Even Calvin cycle, the C3 cycle was worked out in the 50s. Okay, late 50s. And so, you can see that we are talking about, about 60 years, that kind of time scale. All right. Now, these were the two people, Hatch and Slat. They unraveled another mechanism, and it is known as the C4 cycle. Okay. What was the definition of this C3C4? The number of carbons in the molecule, first molecule that is formed, which is a stable entity. Okay, so in that case it was phosphoglycerate, which was the stable entity, and it's a 3-carbon molecule, so it was C3. In this case, the stable entity is this molecule, right, which is known as oxaloacetic acid. So you can think of this part, if I had CH3 here, it would be CH3CO2H, that is acetic acid. So it is like a substituted acetic acid, and this part, is derived from oxalic acid. Okay, and so that's why it is called oxaloacetic acid. Okay, now let us try and understand what exactly was the motivation of the plant, to do this. Okay, the motivation was that the plants are not giving carbon dioxide from the atmosphere to the stroma where the dark reaction is taking place. Okay, the plants rather are supplying a more concentrated form of carbon dioxide. Okay, and so let us try and see what is that concentrated carbon dioxide. That is this molecule, malic acid. Okay, malic acid is a molecule. Obviously, if I make 1 mole of malic acid, it will have 1 mole of carbon dioxide that it can release. This molecule gives up carbon dioxide. It gets decarboxylated, and that decarboxylation takes place in the site of the dark reaction. So the dark reaction in terms of Rubisco, etc., that is not changing. That is the same reaction. Okay, but the manner in which the carbon dioxide was delivered was indirect. Rather than it coming, diffusing from the atmosphere, it is actually getting released by this molecule, malic acid. Okay, so let us try and find out, what exactly is happening and how malic acid is being generated. Okay, it all starts with this molecule known as pyruvic acid. Pyruvic acid is one aldehyde connected to a carboxylic acid group. Okay, it's a C2. Okay, 1, 2. This is pyruvic acid. Oh, sorry, C3, and there's a methyl here. C1, 2, 3. Okay, so that's pyruvic acid. Okay, now, if those of you who are familiar with organic chemistry, you know that I can write this also in the enol form, which means that this will become an OH and this will become a double bond. Okay, that is known as the enol tautomerism. Okay, so carbonyl will form an enol. Okay, so So effectively, that is what forms it. It first enolizes, okay, and so it is known as phosphoenol pyruvate, okay, the enol form, double bond here, OH here, okay. Now that reacts with carbon dioxide and with water in the presence of an enzyme which is known as phosphoenol pyruvate carboxylate. It's an enzyme. 3 carbon, okay, and it becomes a 4 carbon entity, and that is oxaloacetate. I'll show you in the next slide the mechanism of how it is formed, okay, oxaloacetate. Okay. Oxaloacetate, in the presence of 2 other enzymes, transaminase and malate dehydrogenase, okay, it gets converted into 2 other C4 molecules. One is aspartate, this one, okay. I've shown the acid form, but what you'll form is really the ester form. One part will be bound to a probably to an enzyme, okay, and malate, which is this, okay. The transaminase is really required to convert, right, the OH group into an NH2 group, right. That's what a transamination reaction is. Right? Now, don't go into the details of this. Obviously, there are certain redox processes, that take place, right. I'm not going into the details of it, but to tell you now, when you have phosphoenol pyruvate, this is this molecule, right? What happened? It just enolized, so it formed an OH, and it was an olefin here, and this OH plus phosphoric acid forms a phosphate ester. So it is phosphoenol pyruvate, the phospho group, okay, which came in. This is what this is. This is what is reacting with a carbon dioxide, okay, and getting converted into oxaloacetate, which is this molecule. Okay, let me show you now how that happens. Okay, this is your phosphoenol pyruvate, this stuff, okay. Basically, bicarbonate, carbon dioxide in the form of bicarbonate, comes and attacks this molecule. I'm not going to go into all the details, okay, but there are a series of equilibria, okay, and effectively what happens is that the molecule can be expressed, as I told you, in this enol form, this form, okay, and in over here the phosphate has broken up, so it is a O minus. Now in the presence of carbon dioxide, this O minus moves down the arrow, so this will become a carbonyl, C double bond O, and this double bond over here, it moves, okay, and so it will form a carbon-carbon bond between this carbon and the carbon dioxide carbon, okay. So if it, and this will become a CO2 minus, okay. So ultimately it will end up looking like this. This is your CO2 minus part, this one. This is your O minus becoming carbonyl. This is this, and this one, this, okay, went and bound to the carbon dioxide, and so this part has come from the carbon dioxide, okay, and so now it is a 1, 2, 3, 4 carbon entity, and that is known as oxaloacetate, okay. So that's the mechanism by which phosphoenol pyruvate is getting converted into oxaloacetate. Now, oxaloacetate, as I told you, breaks down into this and this, okay, and it is this that goes and attacks the dark side of the Calvin cycle, okay. So what does it do there? Right. So you can see what happened, okay. I had a pyruvate which existed as phosphoenol pyruvate. It took carbon dioxide from the atmosphere form bicarbonate, and it has been constructed in a way that this must be a bit alkaline, this area. So the carbon dioxide will nicely get mopped up in this area, okay? And that bicarbonate goes and attacks this, as I showed you, and then subsequently, it ends up forming oxaloacetate, and that oxaloacetate is converted into malate, okay? The malate leaks into the dark system. The malate is releasing concentrated CO2 because it's a molecule. I mean, lots of these molecules are getting formed, and each molecule is giving you one CO2. Okay, so I've got a high concentration of CO2 which is now interacting with my Rubisco, and once the CO2 has gone off, okay, again it becomes a C3 acid, okay, and again it will undergo certain rearrangements, etc., okay, and get converted into pyruvic acid, okay, from which again you will form phosphoenol pyruvate, and the entire cycle continues. Okay, that is really the way in which now in the dark cycle you are delivering concentrated carbon dioxide. Okay, now obviously when concentrated carbon dioxide is being given and there is plenty of sunshine, then effectively what is happening is that, this stuff, okay, I can push it up because it's not going to saturate over here. It may go up, so I might be able to go somewhere here. I can, and many of these plants, sugarcane, etc., you'll find that the more light that they are absorbing, a lot of irradiance, they become very productive. Okay, these plants. Okay, it is because of this C4 effect that I mentioned to you about. Okay, so the same thing, let us now read it, what it's saying. C4 photosynthesis, the first product of CO2 fixation is malate in mesophyll cells. Okay, so those cells where CO2 is getting fixed is not the place where the dark cycle is taking place with Rubisco. Okay, this is taking place in the mesophyll cells. Okay, and it does not produce phosphoglycerate like in the C3 plants, but it basically forms a C4. CO2 is released from the malate in bundle sheath cells. Bundle sheath cells are those cells where the Calvin cycle takes place. Okay, where it is fixed again by Rubisco and the Calvin cycle proceeds. PEP is recycled back to mesophyll cells. The phosphoenol pyruvate is regenerated, as we saw, and just goes back. Okay, now obviously these processes, like the phosphoenol pyruvate becoming malate, etc., it requires energy. So that's why you'll see there's an input of ATP, there is an input of NADPH. Where are these being formed? It's being formed in the light cycle. Okay, but in the light cycle, instead of directly putting in ATP and ADP into the dark cycle, all of it, some of it is being utilized basically for this phosphoenol pyruvate to malate conversion. Okay, and so it is making very good use of the light, and it is concentrating the CO2. Okay, and it's possible because the CO2 is possibly forming bicarbonate, and then it is reacting with the phosphoenol pyruvate. Okay, the same thing, I mean, just so that you know the anatomy, it's actually a microscopic picture. The oxaloacetate is being formed, and from there the malate are a concentric ring outside of the bundle sheath. Okay, it's over here, and this is where it's full of chloroplast, and this is where this particular reaction is taking place. Okay, and this is the segregation, okay, which is between these cells and these cells. Okay, and then of course you have also other sites like the xylem and the phloem, and these are basically tissues, which are engaged in transport of, because you need other things, you need water, you need nutrients, you need other kinds of organic materials, etc. Food that is being produced, it has to be transferred maybe into the stem or root or some other place. So those are all being done by these xylem and phloem, okay? And this is your mesophilic cells, and this is your bundle sheets, okay? So this is broadly, the kind of anatomy of the C4 plant, okay? Everybody clear? Everybody's understood?
Yes, sir.
Okay.
Yes, sir.
Okay, now let's look at another very clever trick that nature has been playing, Now, see, uh, when does photosynthesis take place in a plant? When does it take place?
In the presence of light.
In the presence of light. So it takes place during the daytime. Okay. Now, if let's say I have the stomata, the stomata are the places, the pores through which actually gases diffuse. Both will diffuse, oxygen will diffuse, CO2 will diffuse, Now, suppose, like let's say I want to block off photorespiration, okay, and I want to promote effectively the dark cycle with carbon dioxide. A very clever trick can be played. Okay, and second is that in the deserts, these are especially for cactus and things like that. These kinds of plants. What happens is that if you because it's so hot in the desert, and my stomata have to be kept open because I have to allow my carbon dioxide to come in. But that same stomata also is the point from which water will transpire, it will evaporate. It will be released from the plants. So I have a tricky situation where I have to keep the stomata open to allow the carbon dioxide to come in, okay, but also my water is getting lost, which I don't want, okay. So what these C4 plants have done, it's a variant of the C4 plants which I told you about. What it does is that during the daytime, it shuts off the stomata, it closes it. So no CO2 is coming in and water cannot go out because the stomata are shut. Okay? At night, what it does is that when it cools down, it opens the stomata. And when it opens the stomata at night, the carbon dioxide goes in then, okay? And what it has done is that during the daytime, the photosystem 1 and photosystem 2 reactions which generate ATP and NADPH, and that's all I require, So effectively, those are there, and it allows the carbon dioxide to come in, and it says, hey, I can produce malic acid at night, via the oxaloacetate. Okay, because I've got my ATP, I've got my NADPH, I've got my carbon dioxide coming in at night. I don't need light anymore. Light has done its job. It created the ATP and ADP. And so, and what am I doing? Even during the night, the dark phase, when otherwise, it's all useless and a plant only respires, but not these plants. These plants get busy. They start working and they start converting carbon dioxide into malic acid. Okay? And that malic acid is stored, and released into the carbon dioxide. Okay? And so, and at that time the stomata is closed, so that the water doesn't escape, and I don't need the carbon dioxide coming in because carbon dioxide came from my malic acid. So this is how plants have adapted, okay? And so you can see, how it is possible to conserve the transpiration, minimize loss of water. You can cleverly make use of 24 hours a day, in basically carrying out effectively photosynthesis, at during the daytime by generating ATP and NADPH, and at night time by grabbing carbon dioxide and making use of the NADPH and ATP to make oxaloacetate and malic acid thereon, and that then goes, into my dark cycle, Kelvin cycle. All clear? Everybody understood?
Yes, sir.
Okay.
Yes, sir.
All right. Okay. Now, the fundamentals of photosynthesis, but not of the downstream products, which we will cover. That we have covered, and anyone who wants to obviously know it in much greater depth, you should go and read up, and I'll also send you some literature, and you'll get more information. Okay. And remember that the materials which you are producing, like let's say I've written down over here all kinds of things: fructose, glucose, sucrose, starch, cellulose, hemicellulose, lignin, amino acid, lipids, organoids. So these are the primary metabolites of the plants. Okay, and all of these they can be combusted. Okay, and I can get energy out of them. That's the heat of combustion. Okay? So what does it mean? That it actually converted solar energy into chemical energy. Okay, and all these chemicals are at a higher energy level, and they can be, burnt and again release carbon dioxide, and you extract that energy now as heat energy. Ultimately, solar energy became chemical energy. Chemical energy you can convert into heat energy, or you can do other things with it. Okay. Now, if you look at the heat of combustion, okay, per kg, if you look at these fructose, glucose, sucrose, you can see, they're all comparable, between 15 and 17 megajoule per kilogram. Okay. Can anyone tell me? Okay, well, maybe I'll go a bit more and then ask the question. Now, when I go to lignin, okay, my heat of combustion has increased. Okay, and when I go to lipids, my heat of combustion has increased further. That means these have got more energy. Can anyone tell me why the energy heat of combustion is more here? Why I am able to release more energy from a lipid as compared to fructose or glucose? Would the oxidation states of carbon be the same in the lipid and in the sugar? What is it in glucose? Say C6H12O6. Carbon.
It will neutrals. I think it's really zero.
Wonderful. When you've got a lipid, it is like what? It's CH3, CH2, CH2, CH2. I can treat it almost like a repeat unit of CH2, the bulk of it. Okay. And so what would be the oxidation state of CH2, carbon in CH2?
-2.
Yes, -2. Okay, so what happens is that as you have carbon in the lower and lower oxidation state, the calorific value goes up. In other words, the heat of combustion is higher because, you see, I can treat a glucose or sucrose, etc., as if it is partway to combustion, of a fat into a carbon dioxide, as if it has gone halfway down the line. Okay, and whereas if I take something like a lipid with a -2 charge, so obviously when it is combusting, it is going to form CO2 plus it will form water, and I'll get the maximum heat of combustion for that. So remember that lower is the oxidation state of the carbon, the more will be the heat of combustion. Okay, and at the extreme would be carbon dioxide, which is +4. Obviously, I cannot combust carbon dioxide; it's already at the lowest energy form. So remember that combustion process always takes carbon from a lower oxidation state to a higher oxidation state. Now let us look at, we have seen how glucose and fructose were synthesized, all right, and we found that it is basically the 3-phosphoglyceraldehyde, and those are the ones, two of those come together, and that forms glucose, fructose, and those C6 kind of carbons. Okay, and now sucrose is a disaccharide. It is basically a dimer of not a dimer. Yes, I mean a dimer of glucose and fructose. Okay, so there are various. So what nature does, is that it takes the monomer units, which is glucose, fructose, etc., and it has evolved a technique by which it can transfer one to the other, and so a monomeric system becomes a dimeric system. Okay, so there are three ways nature actually does this, and they differ for whether it is bacteria or higher plants. Okay, a bacteria, what happens is that the glucose, you remember the primary thing was not glucose, it was glucose phosphate. That's what was formed. Okay, and it combines with the fructose in the presence of an enzyme known as sucrose phosphorylase. Okay, and that's how bacteria make sucrose. Okay, a glucose 1-phosphate combines with the fructose. Okay. Higher plants do it a bit differently. Okay, they have an entity which is known as uridine diphosphate glucose. This is uridine, okay, and this is your glucose entity. Okay, now uridine diphosphate glucose finds an acceptor, which is fructose, and transfers the glucose from here to the fructose. Okay, and it ends up with sucrose, and it ends up with uridine diphosphate. That means minus the glucose. Okay, that's one way it does it. Okay, and the second is that there is another, where you can take the reverse, which is that you can get both UDPG and fructose phosphate, okay, together. Both are in the form of a phosphate ester, okay, and they react with an enzyme in the presence of an enzyme called sucrose phosphate synthetase, okay, and it ends up effectively forming the sucrose phosphate, okay, and then the sucrose phosphate gets hydrolyzed and releases the phosphate, and it forms sucrose. So either you can have one phosphated glucose or fructose and combine with another, or you can take both of them in a phosphated form, both the glucose and the fructose, and in presence of this enzyme, it forms sucrose phosphate and releases the glucose-free uridine diphosphate, and this subsequently gets hydrolyzed and forms sugar. Okay, so that is how sucrose is produced. Okay, now let us look at, because from biomass point of view, Now sucrose, of course, as you know, is very, very important from the uh, biofuel point of view. Where do we use it? Which biofuel we get?
Ethanol.
Yeah, ethanol. I mean, earlier ethanol was from molasses. Molasses is what? Molasses is leftover sucrose plus some glucose and fructose, which are there, plus some salts, etc. And from that you ferment and you make ethanol. Or today you can directly take sugarcane juice. Sugarcane juice is primarily sucrose, and you can ferment it and make ethanol. We also need to know, all right, how starch is made. And later on, we'll also come to know how other things are made, like cellulose, like fatty acids. Okay, so let us look at how starch is made. Now, to understand how starch is made, and this is very crucial that you understand when you if somebody, for example, some of you may be knowing or you may not know, but like if, for example, I want to take, say, potato starch, okay, and I want to ferment it and make ethanol out of it, okay, and it's quite simple to do, okay. And so, for example, when we have a lot of rotten grains, I mean, all the grain we don't consume, many things rot in our godowns, in silos, you'll find there will be all kinds of starchy things like rice, or potato, etc. And these are rotting. So instead of letting it rot, you can convert it into biofuel. But if you, for example, want to make ethanol from cellulose, from the leaves, etc., as such, there's not much of a difference in terms of compositional aspects, but it is very hard to convert cellulose into ethanol. So it is necessary for us to know what is fundamentally the difference between these things. Okay, we should know what is the difference. Okay, so let us try and understand, from these from the slide, okay, some of the nomenclature. All right. Now, firstly, this is like glucose, All right. I can write it as you start the nomenclature for numbering the carbons is that the one with the highest functionality. In this case, the highest functionality is carbonyl. Okay. Acid, carboxylic acid is treated as the highest functionality. Then comes carbonyl. Then comes alcohol. Then comes CH and things like that. Okay. So carbon is treated numbered as 1. Okay. And then I go in sequence 1, 2, 3, 4, 5, 6. Okay. So I've got my numbering over here. All right. Now, what happens is that when these things actually exist, in nature, they don't actually exist in this straight chain form. Okay, they exist in a cyclic form, right? So, for example, if I and what is that cyclic form? I take I enolize this, or I make a hemiacetal. Okay, so I can have this oxygen coming and going and binding to this carbon, and this will then become OH. Okay, so it will look something like this. Okay, so like I've taken a this carbon, which is this carbon. Okay, and so this is number 1, 1, 2, 3, 4, 5. Okay, there is 5, this 5, and then I've got a CH2OH sticking out. Okay, so this carbon and this carbon are effectively combining. Okay, that's what is happening. All right. Now, when I do that, right, so this is my numbering. So 1, 2, 3, 4, 5, and then this is 6. Okay. Now, if I were to form, a straight chain, okay, one way, and this is what nature typically tends to do, that it combines the OH from one, that means from this part, okay, this one, okay, the OH, okay, and it combines the third one, which is this one, this OH, okay, and it forms a oxygen bridge, which is known as a glycosidic linkage, okay, that is a glycosidic linkage. All right, this one. Okay, so this you can see, this is number one, right? This is this one, number one, right? 1, 2, 3. So this 3 and is combined with the 1 from here. So I formed a glycosidic linkage, and this is known as a beta 1,3, glycosidic linkage. Okay. Now, that is not the only linkage. You could also have, this CH2OH over here, right, linking up with one of another glucose, okay, and I form another glycosidic linkage here, and this will be 1,6. That means this is the 6th carbon, right, this one, which was sticking out from 5. This one, okay, this one's oxygen and this one's oxygen. This one is number 1 carbon, okay, this is number 6. So this linkage is the 1,6 glycosidic linkage, and this one is the 1,3 glycosidic linkage. Now, what is the difference between this? If I look at the structure, what is it? What do I see? Suppose I continue like this, okay, what will I see? What do you see?
There will be branching of the
Yes,
chain.
Great, I'm so happy. Okay, so you can see that 1,6 linkage leads to a branching. Okay, whereas 1,3 linkage, if I were to continue, I would get a linear polymer with no branching. It would just continue like this. But starch and many of these polysaccharides, they invariably exist, or they invariably exhibit branching. Okay, there will be some linearity and some branching. Okay, so what this molecule that I've shown you is and I'll explain to you what is beta and what is alpha. Okay, this is stereospecificity. I'll talk about it. And 1,6, etc. What it is regiospecificity. That means which region of the molecule is getting attacked or is combining. Okay, so that is region. So these are regions of the molecule. Okay? Within a particular carbon, if there is stereospecificity, that means it can appear in more than one shape. That is known as stereospecificity. Okay, so we will discuss stereospecificity. Okay, so basically you have, in this particular case, there is an example, a beta 1,6 glucan molecule showing how carbons are numbered. The terminal saccharide is linked via a beta 1,6 glycoside bond. Okay, why terminal? Because this is what I'm doing. This is the last one. Say I'm doing a branching. Okay, the remaining linkages are all beta 1,3. Okay, so this can go on. Okay, here also it can go on. There's no issue. I can form a beta 1,3 with the other one. This OH will combine with another one here, and so I can continue like this, and these would be my branches. Okay, now, so I've explained to you the nomenclature. What happens is that starch is basically a linear helical amylose and branched amylopectin. So let me explain to you now. Okay. Now, as far as this is concerned, the amylose, the only difference is that whereas here you saw that one was being connected to three, 1, 2, 3. Now in starch, the one, okay, this one is 1, 2, 3, 4. The fourth carbon, that is this carbon. The fourth carbon is combining with this one from another unit, right? The number 1 carbon. So it becomes a 1,4 linkage, right? This 1,4 glycoside is this molecule known as amylose, okay? Otherwise, it's pretty much, it's the same as this, except that it is connected as a 1,4, okay? Now, the 1,6 part is the same as here. That is, you know, like we showed over here, the 6 over here. So you can see that now you have got your 1, 2, 3, 4. Okay, 1, 2, 3, 4. Okay, and then this is your 5, right? And then this is your 6, right? So this 6 is connecting with the 1 carbon's OH, the hemiacetal, and that is your 1,6 linkage, exactly like here. So you have a 1,4 linkage, you know, and you have a 1,6 branching linkage. Okay, that is starch. Now the other thing is that the amylose is straight chain, but it is not just linear, it is twisted in the form of a helix. Okay, so there's a helical kind of a structure. So there's a chirality, you know, to this structure, right? And but otherwise it's pretty much the same. And this is starch. Now this is fairly easy to break down, you know, and that's why, you know, starch is after sugar, starch is preferred as a raw material, and that's why many of the grains, etc., they are thinking of producing ethanol now, you know, from rotten grains, because starch can be easily broken down. Okay, so this is starch. Is everybody clear about the structure of starch? The helical
Yes.
amylose and branched amylopectin. Okay, so this part is called amylopectin. Okay, and this part is amylose. Clear?
Yes, sir.
Okay. All right. Now let us try and just understand the alpha and beta nomenclature. Okay. Now notice over here it says alpha 1,4. Okay. Okay. And here it was, I mean, the example that they're given is beta 1,6. Okay. Now you will see that what is happening over here is, if you take the this, you know, the carbon which is number 1, right, and you take the carbon, all right, which is at 5 position, you know, the 1 position and the 5 position. 1 position has the CH2OH group, okay, and the 5 position, this is the hemiacetal part, you know, which forms your glycoside linkage over here. All right. Now, if these are on opposite side, okay, you can see they're on opposite sides of the ring. This is known as alpha. If they're on the same side, it is known as beta. Okay. Now, look at this. When alpha glucose molecules are joined chemically, you get starch, you know. So if it is alpha, that means one like this and one like this, you know. Okay. And when beta are joined, that means when they are on the same side, okay, you end up getting cellulose. And there's a world of difference in the properties of cellulose and starch. Okay? But you can see how critical the stereoisomeric orientation is. Now, you'll be able to, if I show you this, right? Like, for example, what do you see here? You see that that we are talking about, this molecule, right? Say, I've got the I've got the this this one, right? Now, when both of those are shown, okay, like this, right? What it means is that this is beta because it's on the same side. Okay, if one of them had been dotted, it would have been on the opposite side. All right. So this is a beta 1,3 linkage. This is a beta 1,3 linkage, and this also you can see same side, same side. So this also is a beta 1,6 linkage. Okay. Now in starch, as I told you, it's on the other side. One is CH2H over here, okay, and the O over here is on other side. Okay, this OH. So this is an alpha linkage. All right. So it is alpha 1,4 glycosidic linkage in amylose, and whereas this is a beta linkage, as I've shown it here. All clear? Alpha and beta stereo specificity.
Yes, sir.
Okay. All right. Now, just like we saw that how sucrose is formed, that you have got a glucose 1-phosphate, and that transfers to a fructose. You actually more or less repeat that whole process, one after the other consecutively. Okay, and you will end up with amylose. Right? That's effectively what happens. So there's not much of a difference between this and between the sugar that I showed you. Okay, and remember another thing, that some of these conversions are energetically unfavorable, and so wherever it's energetically unfavorable, you'll see, for example, that ATP is getting converted into ADP. That means if I have to take fructose and I have to make it into a fructose 6-phosphate, okay, I actually require energy to convert ATP. I mean, ATP is converted into ADP. That energy is released and it forms fructose 6-phosphate. Okay. So likewise over here, you will find ATP, ADP. From the arrows, you will know exactly whether it requires energy in this particular process. And you can have a look at it in your spare time, but the basic enzymes that are required are again the same. You can see uridine diphosphate glucose, like we had seen for sucrose, and when the glucose goes off, you are left with uridine diphosphate. So it's nothing different. It's the same kind of thing. Okay. Some of the enzymes are different. I mean, this is a enzyme special to starch, whereas that was for sucrose, but the basic chemistry of how exactly these are, a donor is given into the acceptor are effectively the same kind of chemistry. Okay, there's no major difference between the two. Okay, now let us look at cellulose. Now, cellulose, as I said, one of the things I told you is that it is just like glucose. Again, it is a 1,4 linkage, but now it is a beta. That means both of those were in the same direction. Okay, so that is the only difference. This beta, in other words, it's this one. Okay, it's both are like that. Okay, and that is what is combining. And so that's how you make cellulose. But there's one more thing to making cellulose. Okay, one glycoside linkage at the end is formed with a steroid molecule. Okay, this is a steroid. Okay, so this is like beta cytosterol glucoside. Okay, so this is a steroid. Okay, now steroids have a very interesting directing property. Like for example, many steroids you can make liquid crystalline polymers out of it. It exhibits liquid crystalline behavior. So because of this directing property of the steroid, the cellulose is much more crystalline compared to starch. Starch is an amorphous molecule. It has no order, whereas cellulose is highly ordered, very, very ordered. And because of this ordering, what happens is that cellulose forms an extremely compact structure. And because of that compact structure, it becomes very difficult for other things to penetrate. Like if I have to hydrolyze with an acid or an alkali, those acid and alkali can't penetrate into the cellulose because it's so tightly bound. And that tightly bound is coming because of its crystallinity and tremendous ordering affected by effectively the presence of the steroid molecules. Okay, so that's the fundamental difference between starch and cellulose. Starch is basically an alpha linkage, cellulose is a beta linkage, but otherwise both are one-fold linkages, the linear part. Cellulose has a steroid as a directing group, and it forms a much more ordered crystalline kind of a structure, which is extremely difficult to penetrate. And that's why it is so difficult to process lignocellulosic biomass, for example. Very, very hard. And at least this chemistry will at least help you to understand why A behaves in a certain way and B behaves in another way and C behaves in still another way. Okay, it's all because of this. Okay, let us now look at the formation of lipids. Okay, because lipid, as you know, is another very, very important thing. And in fact, a lot of plant lipids, like oils, those have been used for making, besides all cooking and making lubricants and all kinds of things. I mean, they have been used to make biofuels, biodiesel. It's made with lipids. Okay, so lipids are again very, very complex mechanisms. Now, each of these, I mean, what I've shown you in one slide, let me tell you that two people got the Nobel Prizes, for their work related to lipids. Okay. The first was this person, Lippmann, in 1953. He was able to isolate this enzyme known as acetyl coenzyme A. Okay, it's one of the most important enzymes in plants. Okay? The second is that there is these people, all right, Conrad Bloch and Lyman, right, Leinen, they were awarded the Nobel Prize for their discoveries linking acetyl coenzyme A, that means this enzyme, with the lipid fatty acid metabolism. Okay, so and that is what these people effectively got the Nobel Prize for. So what this slide shows you is two things that can happen. One is how I can assemble a lipid from small molecules like glucose getting converted into pyruvate, okay, and then these getting assembled here to, and this part, this business of forming fatty acids is known as lipogenesis. Okay, alternatively, you can feed this stuff, you can feed these fatty acids, okay, and you can metabolize the fatty acid and you can generate citric acid, okay, and it completely decomposes, your fatty acids, and the reason why it releases a lot of energy is that process, which this is known as the citric acid cycle, in the citric acid cycle, the acetyl group, okay, so it's acetyl coenzyme A, this acetyl group, which is CH3CO, okay, it gets oxidized, right? Finally, it forms citric acid, okay, and in the process, it releases 11 ATP molecules. So huge amount of energy, is released, okay, in the process of metabolizing lipids, okay, and that happens in the plants because of this cycle, right, which is the fatty acid metabolism cycle, right? In the other case, what happens is that I take glucose. Glucose is converted into glycerol. Okay, don't worry about how it is converted. Okay, let's not worry about that. And in a similar way, okay, I have fatty acids which were generated here, and these fatty acids in combination with acetyl coenzyme A, okay, these two get together, right, and they end up forming your triglycerides, the triacyl glycerol, which is basically your triglyceride, okay. Now there are also certain phospholipids which can form, right? So, and that comes from the phosphatidic acid, that is, if you don't have a simple fatty acid, okay, but if it is phosphated, you'll end up forming a phospholipid. These sometimes create a lot of problem while you're processing for biofuel. You have to get rid of phospholipids, otherwise they interfere and create a lot of problem in the final running of these vehicles, etc. Okay, so effectively, so that's what happens. Okay, that glucose can get converted into glycerol, or glucose can get converted into pyruvate acetyl coenzyme A, which then undergoes a lipogenesis, and I'll show you how this happens. Okay, and then it's activated, it forms a fatty acyl coenzyme A, it binds to this and the glycerol and this, they esterify, okay, and I can either form the triacyl glycerol, which is the simple lipids, or if it is a phosphated glycerol, it will end up forming phospholipids. Okay, that's basically what it is. And these again can also be hydrolyzed back to glycerol, back to fatty acid, and so I have a continuous cycle. Depending on what I'm doing, I can push the equilibrium one way or the other. Okay, and so this whole process is the lipid metabolism pathway, which is so crucial. For example, making vegetable oils that go into biodiesel production. Okay, and remember another thing, that in all of these cases, right, you will either require ATP and NADPH, for assembling these things, or when you're breaking them down, you can release ATP, as in this citric acid cycle. All right. So broadly, okay, you should get some idea of how exactly a lipid is made and what are the primary building blocks, from which the whole synthesis is carried out. Is it clear to everyone?
Yes, sir.
Yes, sir.
Okay, now let us just look at a little more at the lipogenesis part, which is this part. Oh, by the way, another thing that I didn't mention, it's so versatile, this acetyl coenzyme A, and it's kind of products with pyruvate and all, that you can end up with all kinds of products, lipogenesis, or I can make cholesterol, and from which I get steroids, and those are the steroids which go over here into directing the formation of cellulose. Okay, or I can end up forming ketone bodies. So many different pathways which are basically at the heart of it, which is acetyl coenzyme A, for which we got the Nobel Prize. Okay. All right. Let's see what exactly is happening. Right. It's a very interesting process. Now, this acyl group, the CH3CO group, that is the acyl group. Okay. This is actually bound to an enzyme, okay, which is the coenzyme A, all right, and effectively this is one component. Okay. Similarly, you have the malonic acid. This part is malonic acid. Okay. Malonic acid similarly can be bound to coenzyme A, right, and the two of them are basically now reacting. Okay. How are they reacting? What's happening is that from the malonyl, one carbon dioxide is being kicked off. So I'll be left with a CH2 group here. Okay. And this CH3CO, now this goes and attacks this carbon. Okay. And so it becomes CH3CO from here. And then I've got my CH2, this CH2, and then I've got my COSACP, right? And the other part just went off. The coenzyme went off, one of the coenzymes, and I released carbon dioxide from this decarboxylation. Okay, so I end up forming this. Now, NADPH comes into action. What does NADPH do? It's a reducing agent. Okay, and the beauty of NADPH is that it can reduce both carbonyl groups as well as it can reduce olefinic groups, and both are used in this process. What's happening? The NADPH is going and reducing this carbonyl. Okay, and so it has become now CH3COH H and connected to this CH2. So this is just carbonyl has become OH. That's all. Okay. Why? Through the reduction with NADPH. Now, because I've got a hydrogen here and I've got an OH here, this can dehydrate and release a molecule of water, okay, and I end up forming a double bond. Okay, so this went off as H2O and I've got CH3 double bond CH and then this term. Now another NADPH, again comes in because NADPH can reduce both olefins as well as carbonyl, and it just reduces it to the straight chain. Okay, so now what did I do? Instead of having a CH3CO, S group, I now have a CH3CH2CH2COS group. So I introduced two carbons. Okay, where did I introduce those two carbons from? From my malonyl. All right. Now, next time, this will not go and react with this. This will go. Okay. So now instead of forming this, I'll have CH3, CH2, CH2, COS, ACP. That's the only difference. Now, again, it will go and decarboxylate this and go and attach here. Okay. And each time I'm increasing the carbons by 2. And each time I'm requiring 2 NADPH, one to reduce carbonyl, okay, to OH, and the other is to reduce olefin to saturated. Okay, that's effectively what I am doing over here. And you keep building this up, and typically, the fatty acid side chains are anything between C12, like let's say in coconut oil, to like lauric acid, to something like C23. So you can have fatty acid with different chain lengths, but the fundamental process is effectively what I am showing you here. Has everybody understood how lipids are formed?
Yes,
All
sir.
clear?
Yes, sir.
Okay.
Sir.
And any question?
Yes, sir.
No, any question from anyone?
Yes, sir.
Yeah, tell me.
Where can we get this malonyl group?
That's a very good question. I'll have to see, just like you have pyruvate form, okay, there is a pathway for also making malonic acid. Okay, I have not shown the pathway here,
Yes, sir.
right? Maybe I will, you can look it up yourself, what is the malonic acid pathway, or maybe next time, or in the, along with the material, I'll try and dig it out and send it to you. Okay.
Thank you, sir.
Very good question. All right. Okay. Now, if you also look at, this whole process, all right, I've tried to summarize for you, that what are all the fatty acid synthesis enzymes, and by the way, all of this material, it's given in very lucid manner in Wikipedia from where I have taken the thing because I found them explaining it extremely easily, So all of these enzymes, what they are, what is the reaction that they do, okay, and effectively everything, right? You'll be able to, and what is exactly the function of the enzyme, you'll be able to read from here. And so it basically just repeats, whatever I have told you already. Okay.
Yes.
You can see like enoyl ACP reductase, okay, reduces the C2C3 double bond. Okay, so all the things that I over here, this one, right? It carries out the dehydration. This one, it carries out the reduction. All right, all those. So you can see this was the dehydration step, is carried out by this enzyme. This was the hydrogenation step, last step is carried out by this enzyme. This was the reduction of the carbonyl, is carried out by this enzyme. Okay, this is the chain extending. How the CH2CO group transfers from one to the other. That's carried out by this enzyme. All right, and so this is and the malonyl. How it is activated. That is this enzyme. How the acetyl is activated. It is this enzyme. Okay, so it's all very, very nicely explained. All right, and these are the transfer kind of proteins which are there. Okay, the ACP is the acyl carrier protein, and this is the coenzyme A. All right. Okay, now let us. We have now covered the fundamentals of photosynthesis and biosynthesis. Okay, now we will come to actually making use of biomass. All right. So the rest of today's lecture and including the maybe the next lecture and the lecture after that will be all focused on how actually the biomass is getting used. We have identified the source, but how it is being really converted into a required renewable energy form is what we will look at, over the next maybe 2.5 lectures. Now, what is very important for you to know, this is not true in India at the moment, okay, but the world is aiming to get 50% of its global primary energy from biomass. This is what the world wants to do. I think I've shown you something similar in one of the previous lectures, and so we really need to understand how to boost this area in India, and so I've just put down a few thoughts. Biomass had a dominant role in India in the past and there is a declining trend since 1971. Okay, the reason is, because fossil fuels it started proliferating everywhere. And so even people who were collecting twigs and burning them, they switched over. And I mean, there's nothing wrong. I'm not saying there's anything wrong in doing that. After all, we're all doing it. But the fact is that those things are going to come to an end. And if any of you watch the news today, I don't know if you know about the United Nations session that is taking place, and whether you have listened to any of these speeches by the United Nations Secretary General, by Biden from the US, by the Prime Minister of UK, and many others. Everybody is focused, on really trying to get rid of fossil fuels, and especially coal, and really going more and more for renewables, and all because we have to contain the temperature rise to less than 1.5 degrees C. The temperature has risen so much that there can be massive flooding. It's already happening. There are many different scenarios which are playing out. We'll have to look at those. Okay. So to arrest the trend and thereafter to reverse it will require a dramatic shift in mindset, reformulation of policies, and working steadfastly towards set goals. That's what you all have to do. Because people like us have very little role to play now, because we have served our innings. Okay, and you all will have to take things forward. It will also require the most appropriate and efficient technologies in keeping with modern times. Okay, so some things will be the same, but not quite the same. I mean, after all, knowledge has progressed, technologies have progressed. So you may increase the use of biomass, but how you actually utilize it and what technologies you deploy could be different. Okay, and that is how science and technology moves forward. Biomass can be converted into desired forms of fuel and energy, depending on the nature of feedstock and conversion processes. Okay, now this is something, and increasingly as we go through this course, I hope some of the basic logic will become clear. Like, for example, let me go back to one of the previous slides. Yeah, like, for example, I told you that lignocellulose is very difficult to process. Okay, I mean, so if you, for example, want to break apart the chains, hydrolyze, get sugars out of it, then ferment into sugar, I mean, it's a difficult task. Okay. On the other hand, you see its calorific value is quite good, 26 to 30, and I would rather burn it, and generate thermal energy from it. Why would I? Because I don't have to do anything. I just have to literally light a match, and it will start releasing energy as it burns. Okay. On the other hand, if I've got lipids, now lipids would be ideal to convert into biodiesel. So there, I'm not going to burn the lipid, okay, like this. Okay, I would much rather, try to make biodiesel out of it. And if I'm into sucrose, glucose, fructose, etc., starch, which are already hydrolyzed or very easy to hydrolyze, what I will do is that I'll make ethanol out of it, because it's easy to make. So how you use these things will really depend, okay, on what is your plan, what do you want to do. Okay, and so keep this in mind. And the most foolish thing, right, is to try and convert everything into biofuel, into here, because the energy penalty, the capital investment, etc., simply cannot justify it. Right? So always try to do the smartest things with the appropriate feedstock. Many new conversion processes have been developed and are being developed continuously, which will no doubt facilitate a large role for biomass energy without compromising on convenience of use. Because I mean, I'm not asking you to compromise on convenience of use. I'm just saying use the right thing for the right purpose. Okay? All such technologies may not be applicable in all situations. Other factors such as availability of resource, economic condition, environmental impact, etc., are equally important. You'll have to keep that in mind. Also remember, if you just go and start chopping up forests and saying, hey, I've made biomass, that is as good as non-renewable energy, because you may not be able to regenerate the forests in 2 years. It takes time. And so the rate at which you are utilizing biomass must be matched by the rate at which new plants are coming up. And if not new, not only coming up, it's increasing. Okay, so in fact, I used to see this in Brazil. They keep clearing trees and to make chopping down thousands of trees to make toilet paper. But every time they cut down a tree, they also plant the tree, and it's all very well worked out so that the new plants start coming up. Okay, you have to look at those aspects. There are 4 main products which can be derived from biomass, 2 related to energy. One is power and heat, okay, and the other is liquid fuel for transportation, okay, and 2 related to chemical feedstock and materials, because there are so many kinds of chemicals, okay, and materials. I mean, like whether it's a jute bag, I take jute, I make a bag out of it. Okay, I mean, I can do so many other. I can make so many chemicals. I can take ethanol derived from a plant, and I can dehydrate, make ethylene, and make polyethylene out of it. So I can make chemicals from natural biomass, or I can make materials. Like jute bag is there. And so effectively, that's really what one ends up doing with biomass. And conversion of biomass to energy, chemicals, and materials includes different process technologies, and which we are going to discuss subsequently. Okay, and they could be just thermochemical techniques, they could be biochemical techniques, okay, they could be mechanical techniques, mechanical extraction, okay, or they could be simple chemical treatments, okay, not thermochemical but chemical. And these are broadly the types of processes by which biomass is converted into useful products. Okay. All right. Now let us just look at a couple more other things. I mean, when people started talking about bioenergy, they started talking as if there was no use of bioenergy before, and so they named whatever they were capable of doing, like taking sugar and fermenting to ethanol, and they called it as the first generation technology. Okay, so G1 technology. Okay, but they forgot that long before that, I mean, our villagers they did not know any technology, but they were using biomass. Okay, I mean, they were going and collecting them. They were taking it and lighting up their chullas. Many people did other things with biomass or bioenergy. Okay, now one thing is there that when they do these, you do not know all the implications of what you're doing. Like, for example, if I'm burning biomass in a chula, I might be generating certain toxic vapors. Okay? And as a villager, I don't know all those things. I only am taking advantage of the good things like thermal energy. But suppose it is releasing dioxin into the atmosphere and I can get cancer. Who knows about it? I mean, they don't know. Okay, second, they have not been bothered about whether you're over harvesting your biomass and denuding your forests and green patches. So some of this collection activity can be very predatory, because you're like a predator. You're grabbing things and taking it away and eating it. I mean, eating it means consuming it in for something, and you're not bothered about whether you're regenerating it. Okay, so traditional things had many good things. Technologies were very simple. Okay, nothing to do specially. Okay, but they also had a downside. Now, when we talk about innovative technologies, and we are talking about first generation, second generation, third generation, etc., I will call this as zero generation, traditional. Okay? Now, here, what you are supposed to do, whether you do it or not, is a different matter. Typically, you have to look at biomass as a agro industry where you are actually utilizing planted material, right? And you are recovering biomass from planted material. So you are recovering biomass from planted material, and also you will replant more plants so that those will again grow and not grow over millions of years. That those have to grow within 5 to 10 years, and you have to reach a steady state, so that the rate at which I'm harvesting biomass and the rate at which new biomass is being created more or less will match. Okay. Now, whereas here most of it was for thermal energy, today I could look at uses in terms of transportation fuel, okay, liquid fuels, okay. I could look at it for generating electricity, power generation. I could look at it for agro industry, okay. So I can now look at it in a much broader way than what was possible with traditional industries, okay. Now, conversion technology, I mean, they are simple as I told you. I mean, you just burn them, okay. Um, or even if I make materials out of it, I will do it in the crudest of manner. Okay? But I don't know. Nobody has worked out energy. If you go and ask somebody, hey, tell me what is the energy efficiency of your process? You don't even know the calorific value. You don't know how much energy was formed. What energy efficiency? I mean, they're just getting some use out of it. Okay. Whereas, now when you are looking at it in more innovative ways, I will started looking at efficiency. Which is the most efficient thing to do? What is the most integrated way to do things? I'll start looking at all that. Okay. Now, in terms of traditional, as I told you, I mean, people just went and collected it, from wherever, and they took it and used it. Nobody asked them to pay anything. There were no prices, okay, for this. Whereas today, I mean, even if let's say in suppose in Haryana, that when they burn their crops and it generates a lot of smoke and all of Delhi is suffocating. But if you go and tell those guys, hey, I want to pick up your biomass, will you give it to me? The guy will not give it to you for free. At that time, what otherwise he might have said, ha, leke jao, moment he sees that, array, these guys are doing something useful and they're going to make money out of it, he'll start charging you. Okay? So then there will be a price. Like, for example, we have had this, there have been technologies developed for making ethanol from lignocellulosic biomass. and all the technology everything looks great, but when you do the costing, okay, and you do realistic costing and say no, even raw materials you have to associate a cost with, I mean all these projects have become unviable. Okay, they don't work. And as I told you, environmental impacts today are absolutely nobody will allow you to do any project on biomass if you do not have a good understanding of the overall environmental impact of your project. Okay, you must know. I mean, for example, if I'm utilizing more energy to convert A into B, what's the point in converting A into B? Okay, and so I might say that no, I'm not going to allow this. It is environmentally unsafe. Okay. So these are the kinds of things that you need to understand. So although modern bioenergy tends to displace the traditional one, this trend should be supported by proper social, environmental, and technical guidelines. And that is where policy comes in. You need guidelines, you need policies, so that people are not doing things randomly. Like, for example, as I said, I mean, today people are converting sugarcane juice into ethanol. I mean, surely that is going to raise the price of sugar, because the sugar that could have been produced is now getting diverted to ethanol. And so one will have to ask, is that the right thing to do? Okay, maybe today somebody will say, no, we have got surplus sugar. Maharashtra is producing so much sugar, we don't know what to do with it. Yeah, maybe, but you will also have times when you will produce much less. And there are so many other factors that you need to consider. Sugar is a water guzzler, takes up a huge amount of water, fertilizer to cultivate. I mean, so you will pay a price. So, I mean, all of these are the kinds of issues, that you need to look at.